introduction, Amy. I'm here to introduce Sethera and some of the enzymatic techniques that we developed at the University of Utah. We are building upon basic science to bring an underdeveloped class of therapeutics forward. In the world of medicine, small molecules have been the heroes of drug development. But what if we told you that there's another class of underutilized heroes that have been waiting in the wings, ready to open new avenues into how we treat disease? Peptides are short chains of amino acids. While they've been part of the pharmaceutical landscape for ages, their immense potential has been largely untapped. Why? Certain peptide therapeutics, such as octreotide, have been used since the 1980s, and peptides have an ace up their sleeve. They can be made to have a level of specificity that's unmatched. They can zero in on specific proteins or receptors with pinpoint accuracy, even to the most difficult targets. This is a boon for tackling those elusible, undruggable interactions. These compounds are precision masters, are capable of orchestrating intricate interactions that open doors to treating previously untargetable diseases. The few peptides that have been brought to market have been blockbuster therapeutics. Octreotide was a $3 billion molecule in 2022. However, this potential is met with challenges. Peptides, by their nature, often suffer from short lifespans in the body, and when taken orally, they face a gauntlet. The journey through our digestive system often sees them broken down and rendered ineffective before they reach their target. The entirety of the human body is hostile to peptides, and the structure of a peptide determines its activity. Removal of even a single amino acid from the peptide can render it inactive as a therapeutic. They are not safe in the blood, nor are they safe within cellular environments due to the specific hostile nature of those areas towards chains of amino acids. One way to introduce stability and increase efficacy of peptides is to cyclize them. A cyclic peptide helps lock critical components into the most effective conformations, shapes, and orientation. This helps protect the peptide from many biological processes as well. A linear peptide is like a flexible key that only sometimes opens the lock and very easily breaks. But a cyclic peptide actually rigidifies that structure and always opens the lock and is safer from breakage. However, the traditional way of generating cyclic peptide structures produces very fragile links. The bonds can break easily inside of the body, neutralizing the peptide's power and rendering it exposed to the body's natural processing. Further, think about this. If one cycle is good in a peptide, two or more is better. Imagine if we could controllably place multiple cyclic bonds in a peptide. It would allow for unique three-dimensional structures that could open even the most elusive biological locks or act as a goalkeeper, keeping specific receptors safe from unwanted activation. This is where Sethera shines. While traditional methods have been used, such as disulfides for peptide cyclization, which have significant limitations, the power of nature is what we use. We harness enzymes, both naturally occurring and modified, in order to process peptides. By leveraging nature's innate machinery, we can achieve what is truly groundbreaking. We can orchestrate complex reactions at room temperature under exceptionally mild conditions and in a one-pot reaction. The results? Biologically stable thioether cycles within peptides, achieved with elegance and efficiency that only nature can provide. While the traditional methods to produce cyclic peptides result in a reversible bond, our process installs an irreversible thioether bond that is stable in biological conditions. Our enzymatic techniques redefine the frontiers of peptide modification. Our processes are capable of installing thioether bonds within a diverse array of amino acid frameworks. Not only can we target side chains, but our enzymes also adeptly handle unnatural amino acids like D-amino acids and beta-amino acids across the entire peptide sequence. This adaptability allows us to modify peptides even when every residue, intervening or otherwise, is unnatural, which is a remarkable deviation from conventional enzymatic constraints. We are not limited by the context of peptides either. We can reliably install thioether bonds in peptide small molecule conjugates in a site-specific and stereo-specific manner. How do we demonstrate the power of these enzymatic techniques and the peptides they produce? Well, the oral availability of peptides remains a formidable challenge. Novartis has recently published results showcasing a peptide that can be taken orally, passively permeates the cells, and this is largely thanks to its N-methylated residues. This represents a significant leap forward in therapeutic design. However, such compounds can be very complex to produce. Sethera's enzymatic technique simplifies this complexity, offering a more streamlined production with higher yields. Our process uniquely tolerates a variety of peptide sequences and only requires a short incubation time with the enzyme, which is testament to its efficiency and adaptability. If we next consider another fundamental challenge to peptide therapeutics, that is the highly reducing environment within cells. This environment often dictates the fate of peptides, especially when cyclized via a disulfide. Glutathione is a powerful antioxidant, and it is incredibly concentrated in nearly all human cells. Some would be familiar with the rapid reduction of disulfides to thiols by cellular glutathione. This not only linearizes the peptide, but also exposes it to proteolytic digestion, which compromises its efficacy. We've pitted our thioether-linked analogs against FDA-approved therapeutics, revealing superior stability and promise. We mimicked the cellular concentration of glutathione and demonstrated that after 30 minutes, essentially all of the disulfide-containing octreotide has been linearized. However, after 18 hours, 98% of our thioether cyclic analog remains. While these preliminary stability results are encouraging, another hurdle exists. Do our compounds show similar receptor activation to already approved therapeutics? And how do they compare against natural disulfide ligands? Our very early preliminary results demonstrate that our compounds are able to similarly activate biological receptors in vitro tests. Here, we pitted several of our thioether cyclized compounds against both the native ligand of SST14 and the FDA-approved therapeutic octreotide in a receptor activation assay of somatostatin receptor 2. Here, Seth131 is able to activate the receptor better than the natural ligand and with similar efficiency as octreotide. The power of our platform is not limited by single linkages. This is critical because controllably installing the intricate architectures seen in important biomolecules like insulin has been a formidable challenge outside of using patented plasmids. But even still, with those methods, they cannot easily use unnatural amino acids. From a traditional chemistry standpoint, issues such as homodimerization, unwanted propagation of one chain or the other are significant problems to overcome. But with our enzymatic prowess, we've established controlled, directional, and specific thioether linkages, opening doors to highly tailored interpeptide chain formation. As a reminder, at the heart of all of these breakthroughs are enzymes, nature's own master organic chemists. What we've achieved at Sethera is the ability to harness enzymes in directing a myriad of specific bond formation events across very diverse substrates. We first install 2 thioether bonds in the peptide substrate. We next cleave at specifically installed residues in order to generate a stable interpeptide thioether linkage. This demonstrates that we can now controllably generate interpeptide linkages using this bi-enzymatic system. Finally, we use chemical techniques to deprotect and oxidize the final bond in the architecture. This allows us to controllably install insulin-like bond architectures in 3 steps. The resulting peptide contains 2 interpeptide linkages and 1 intrapeptide linkage. This kind of architecture is incredibly difficult to install through traditional organic chemistry methods, given how many chemically reactive groups are present in peptides. This is the power of Sethera's technique. What we are demonstrating is a technology that allows for site-specific, directional, with atom-level control of macrocycle size and allows for multiple bond formation events to occur controllably through enzymatic means. Enzymes have been unveiled as the maestros of organic synthesis in our labs, scripting a new chapter in drug development. Sethera is currently at the forefront, orchestrating this dance of enzymes and molecules and crafting the next generation of peptide therapeutics. Thank you.